Over to you, Judith. We're going to do Tuesday's paper first, which was that? Was that Winter 16?
2016? Yeah.
Yeah. Winter 16,
I think.
paper 41.
Yes, I'm going to go fetch it as well.
I'm putting it on full screen. Actually, what you can do, if you zoom in a little bit, so we can see the paper a little bit bigger, can you do that?
Yeah, sure.
There's a setting in PDF Expert to just.
Is that better?
Yeah, it's one more, I reckon. Yeah, perfect. Can you see that kind of code?
Okay.
How good is that? Okay, over to you guys then.
Okay, so welcome. Cool. So do you have any specific issues to target, or should we just start going through it and then as they come?
I think we just start over through the papers.
Okay, sure. Cool. So, oh, actually, there's quite a lot of feedback. Hopefully that won't be a problem with my voice. Anyway, okay, you can just start. Cool. So, DI is a condition that results in excessive thirst and the excretion of large amounts of dilute urine. The main cause of DI is the deficiency of ADH. ADH normally binds to receptor proteins on cell surface membranes of cells in the collecting ducts of nephrons. Outline the effect of ADH on the collecting ducts. Okay, so what were you thinking about this one? So ADH, antidiuretic hormone.
Yeah, maybe like enzymes, like, I mean.
Okay, yeah, so what do you mean by an enzyme? Okay, so what's the normally, if you were to use ADH, what's the effect that has on the collecting ducts? So what's a diuretic? Okay, so a diuretic is like coffee, for example. So if you drink a lot of coffee, that it makes you want to go pee. At least it does to me. If I drink it too late in the morning. Cool. So antidiuretic means that, yeah.
So like maybe like change water levels, like high water potential to low water potential, something like that.
Okay. Yeah. So basically, what it is that if you don't have enough water in your body, that's when ADH is useful, because that means that you can retain more water from your kidneys. So when the whole filtration process happens, then you can, so basically what it does is it makes the collecting ducts more permeable to water. And since surrounding the collecting ducts, it's like the interstitial fluid is quite salty already. So then the water can move down its concentration gradient. So, okay. So one of the ways it can do that is, so normally a plasma membrane is impermeable to water, right? Because so if you have like your phospholipid bilayer, the outside is hydrophilic, and then you have the tails are hydrophobic, right?
Yeah.
So what's one way of getting water across an impermeable membrane? Can you think of anything? Okay, no worries. So like one of the things is in the membranes there is lots of different proteins. So there are proteins that are called aquaporins, in which there's a channel in the middle, which is hydrophilic, so that basically allows the water to go through it. So one of the things that ADH can do is that you have these vesicles preloaded with these aquaporins, and then it can sort of encourage the vesicle to fuse with the membrane, and then the water can go through.
Mm-hmm.
Okay. Does that make sense?
Yeah, it is. Yeah.
Okay, cool. Let's just move on. Okay, cool. So, an inherited form of diabetes may be caused by faulty membrane receptors. These receptors are coded for by a sex-linked allele. Okay, so what's sex linkage?
I thought it's kind of like sex chromosome, but
Yeah, that's good. Yeah.
it makes sense for me. Oh,
Sorry, what did you say?
oh, so it's sex chromosome.
Yeah, pretty much. Yeah, spot on. So, yeah, so your allele is located on the X chromosome, rather than, you know, any of your other 23 chromosomes, pairs of chromosomes. It's specifically on the X chromosome. So I'll just maybe. Sorry, I can't really write because it's with a mouse, so it's a bit hard.
Yeah.
Maybe I'll just leave it as X next time. Anyway, okay.
Okay. Yeah.
Okay, cool. So next, use a genetic diagram to show how parents who do not have this condition can have a child with inherited form of DI. Oh wow, these do have annotations. Let me just take those off. Sorry about that. Okay, cool. Perfect. So, so if the condition is, we know that it's carried on the sex chromosome, right?
Yeah.
And we also know that the parents don't have it. So can you tell me anything about the trait that comes to your mind? So what I mean by that is, how can two parents who don't have a condition have a child that does?
So like I thought like I wrote some diagram like capital F and small capital A and small a. So like they got like big A and small a and big A and small a. You know what I mean?
I think I know what you mean. Sorry, I think you're ahead of me. So what I was trying to get at before the diagram, sorry, was the fact that if the parents don't have the condition but the child does, that means that the allele is recessive, right?
Yeah.
Okay, so that's all I was trying to say is that yeah, so capital A is the normal and then small a would be the DI. Yeah, but you're absolutely right. So that means, you know, that's the mom and that's the dad. And then that's what you said, right? So capital A, small a, capital A, right? Like that.
Mm-hmm.
Yeah, that's perfect. Okay, cool. So the gametes would be each of these four, right? So,
Yeah.
okay. And then what did you get for the offspring genotypes?
Like maybe capital A and capital A.
Yeah,
Like,
perfect.
and then like capital A as well and Y, I think.
Yeah, perfect.
And then
Maybe it helps if we draw a diagram.
yeah.
Okay, let's do that. Maybe with a different color so I don't confuse you. Okay, so here are the gametes for the dad.
Yeah.
And then for the mom. Yeah, so that's the one that you already said. Perfect. These two. Oops, I actually messed it up. Sorry, I meant to write that.
Small A, yeah.
Okay, so that would be,
And small A.
yeah.
And small A and Y.
Okay, perfect. Cool. And so your phenotypes would be this one?
Yeah. Oh, like all of them, I think.
Yeah, so all of the ones that have the capital A would be normal. So, and then only the chance of only one of the males to have DI because this one has the capital A, and you know he has no other X chromosome to mask the effect of the yeah, okay, cool. Do you maybe have any headphones? Just because I can hear myself like sort of. Do you have any headphones to plug in?
Oh.
It's okay if not, I was just. It's just because there's like a little bit of feedback from my voice, and it might be annoying for people who are watching.
Oh.
But it's okay, don't worry about it. I can sort it out later.
Okay.
I'll try and speak more quietly, maybe that helps. Okay, next one. Grass crops such as maize, sorghum, and sugarcane are C4 plants. They're common grass crops of tropical regions. Oats and wheat, commonly grown in temperate regions, are C3 plants. Most plants are C3. They're termed C3 because of the first product of photosynthesis, 3-carbon compound. Okay, so what do you think of this one? Can you think of anything that's just right off the bat that's different between the two biochemistries? So what do you know about C4 plants?
I have no idea what is C4 and C3 plants. Like maybe like carbon dioxide, like number of carbon or something.
Yeah. So actually, that's a really good thing that you said that. So.
Oh, really?
Yeah. So basically, what it is that. Sorry, just one sec. Need to mute an application. There we go. Okay. So. C4 is, there's not a lot of plants that have this sort of photosynthesis, and what happens is it only happens in plants that have a certain anatomy. It's called a Krantz anatomy. Have you heard about that at all?
Not really. Oh, yeah.
No, it's fine if not. Don't worry about it. I was just looking if I can get a picture up, but maybe it's easier if I just explain, I'm not sure. Anyway, okay, so essentially what happens is that the reason why C4 is evolved is because, so Rubisco can use carbon dioxide as its substrate as well as oxygen.
Okay.
And if you are in an environment in which carbon dioxide isn't as concentrated, then Rubisco can be quite inefficient because it can react with oxygen instead. Does that make sense so far?
Yeah.
Okay, cool. Yeah, and so what C4 does to make Rubisco more efficient is that it separates the light independent reactions from air. So what it does is it first, instead of carbon dioxide reacting with Rubisco, it reacts with PEP carboxylase. So it makes a 4-carbon compound. So from PEP, so it's a pyruvate molecule, and carbon dioxide, it makes oxaloacetate or malate. And then that can diffuse into another cell called a bundle sheath, in which the carbon dioxide is released again, and then normal, photosynthesis, as you know it as well, with Rubisco can happen. And the reason why this is good is because you only have carbon dioxide in that environment in the cell, so that makes the whole Rubisco more efficient.
Oh, hello.
Does that make sense?
Yeah.
What were you going to say? Sorry.
No, never mind. That's fine. Sorry.
Are you sure? Okay. Yeah, so you can mention any of... Okay. Yeah, so you can mention any of those that I just said. So, that the first product is a 4-carbon compound, or, the fact that carbon dioxide reacts with PEP instead of ribulose bisphosphate. Yeah, so let's just move on. I think talked enough about that. Okay, so the next one. The C4 pathway for fixing carbon dioxide was worked out in 1966 by Hatch and Slack. During their investigation, they measured the rates of fixation of carbon dioxide at high light intensities in leaves removed from both temperate and tropical grasses. They also measured the rates of activity of two carboxylase enzymes in the leaves, ribulose bisphosphate carboxylase and PEP carboxylates. Okay, so what can you? So these are going to be our C4, and these are our C3 plants. So the first question, I think, is just on compare the rates of fixation in C3 and C4 grasses. So what can you tell me about this column?
So rate of C4 is higher than C3, I think.
Sorry, come again. I couldn't hear you. It's just a bit quiet.
So like mean rate in C4
Yeah.
is higher than C3.
Yeah, perfect.
And should I find like the number of mean rate?
Yeah, you can do if you want to.
Okay.
Yeah.
I don't know. I got okay.
No, I think that's perfect, what you said.
I got one so far, but I need one more. But...
Yeah. I think yeah. So the other one is just basically that you have more variation in these three than in those two.
Okay.
But I checked, and that's already 2 marks what you said, actually, because you mentioned that they are all higher, and then you also brought in an example, so like the mean. So that's was already enough what you said, actually.
Okay.
Okay, cool. Next one. Describe the role of rubisco in the Calvin cycle. Okay.
Yeah. What is this?
So do you have any ideas? So what's the point of the Calvin cycle in general?
Calvin cycle, so it's changed to energy, like in photosynthesis.
Okay. So in order to make your sugars, you need that matter from somewhere, right? So you need your carbon from somewhere.
Yeah, sure.
So what rubisco is amazing at doing is that it can fix your carbon from the air, from carbon dioxide, right?
Yeah.
Okay, cool. And then, yeah, if you, I mean, I'm not going to draw the Calvin cycle here. I think that's a bit ambitious. But essentially, yeah, so ribulose bisphosphate is a 5-carbon compound. And rubisco helps to react your carbon dioxide with ribulose bisphosphate. And that would give you a 6-carbon compound, right? But that's it's already it's not a very stable compound, so it breaks down into phosphoglycerate. So, and the reason why it's 2 is just 2, 3 carbon compounds. So, yeah. Okay, cool. Oh yeah, I should probably write rubisco there. My writing is really not working, is it? Okay, cool. So with reference to the table, suggest reasons for the differences in activity of the 2 carboxylase enzymes. Okay, so here's your enzymes. So is there anything that you can spot?
So PEP carboxylase.
Yeah.
It has higher rate of activity in C4 than the
Perfect.
And also rubisco has higher rate of activity in C3 than C4.
Yeah.
2 more.
So why that? Why might that be?
Because PEP carboxylate is higher, so it's more they need to fix. How to explain it?
So one of the reasons is just an easy one. So maybe C4 just have more of the PEP molecule that it reacts with. So that could be one reason, right?
Yeah, true.
But also C4, they usually live in much warmer and like high light intensity climates.
Okay.
So yeah, so that might be one reason.
Yep.
Cool. Okay. And then actually, so the Rubisco one is a bit more tricky, why it's higher in C3, because the whole point of, C4 is to make Rubisco more efficient, right?
Yeah.
Yeah. So if you have an environment in which you have a lot of light and a lot of warmth and a lot of water, then you might have overall a lower concentration of carbon dioxide in the first place, because you are much more efficient than if you are maybe in like a colder, more shady climate, right?
Yeah.
Okay, so that could be one reason. And another reason is just actually it could just be that the C3 itself is more efficient at higher carbon dioxide concentrations as well. So you could put any of those. Okay, shall we move on?
Yeah.
Okay. It's been calculated that to produce 1 molecule of glucose, the C3 pathway uses 18 molecules of ATP and the C4 pathway uses 30 molecules of ATP. Suggest why C4 plants can afford this high cost of ATP. Okay, cool. So what do you think about this one?
High cost of ATP. Oh, because like it's more efficient. Because why? Because like if it's warmer and like there's light, it produces more.
Yeah, perfect. That's exactly it. You got it. Okay, cool.
Light and temperature.
Yeah. So basically, they can afford to spend the ATP. Okay. Should we go into the next one?
Yeah.
Okay. The polymerase chain reaction is used to produce large amounts of DNA from a very small original sample. The main stages of a PCR are shown in figure 3.1. Okay. So do you know this reaction?
I think so, yeah.
Okay, cool. Perfect. Okay, maybe let's just get into the questions and it's easier to talk through it, I think.
Mm-hmm.
Okay, so first explain why the DNA sample is heated to 95 degrees C in step 1.
So by breaking hydrogen bonds.
Yeah,
And
perfect.
to separate to like two what's that called, strands?
To what, sorry, what did you say?
So just to separate the two strands.
Yeah, perfect. Okay, yeah, you got it. Cool. So that's needed, to have your templates essentially for copying.
Mm-hmm.
And then explain why primers are added in step 2.
Oh, to bind to the DNA, the base painting.
Yeah. Do you have any other ideas?
Any ideas?
So like it binds to DNA, but like where does it bind?
Oh,
It doesn't just bind anywhere, right?
yeah. What is it? I know. I know what it is.
Don't worry.
Oh my god.
That's okay. Don't worry about it. I think you're overthinking it. It's literally just, you know, wherever it finds a complementary site. So the reason why that's would be cool is if you were, for example, at like a crime scene, right? And you found like a piece of hair, then you could, like, you don't need to amplify the whole DNA sample from the whole cell, right? Because you can identify a person from a much smaller set of genes. So if you have a primer for a specific sequence within the whole genome, then you can just amplify that fragment. Does that make sense? Because then you put your primers in that will bind at whatever place you want them to bind at. Does that make sense?
Yeah.
You sure? Okay. Sorry, I would try to draw it if you want, but I'm not that confident in my mouse drawing skills. Okay, and explain why the enzyme Taq polymerase is used in step 3.
Okay.
So polymerase, yeah?
Not sure about Taq polymerase.
Okay, cool. So it's just a DNA polymerase. But why it's special is because the Taq, you don't have to know this, but it stands for Thermus aquaticus. It's a species of bacterium.
Okay.
And they live in hydrothermal vents, so like really hot water. So they have to have a thermostable DNA polymerase because otherwise they couldn't copy their DNA, right?
Yeah.
So the tertiary structure of the polymerase is so that it's stable at much higher temperatures than, for example, our DNA polymerase. You know, by disulfide bridges and stuff like that. So, yeah, it's just added so that it can extend your, copy your DNA sequence.
Okay.
Does that make sense?
I think so, yeah.
Okay, because you can tell me if it doesn't. That's why I'm here. Okay. Okay, should we move on to the next one, or?
So that one is the number 3. The answer is more efficient.
Yeah. So yeah. Actually, that's right. So Taq polymerase could work at 65 degrees C as well, but they've basically just determined that the most efficient temperature is at 72 degrees. So that's why they raise it.
Okay. Yep.
Okay. And then after, you know, you copied your sequence, then step 4 is just basically starting the whole thing over again.
Okay.
Okay. Cool. After an organism dies, its DNA gradually breaks down. However, cells in bones that were buried hundreds of years ago may still yield small amounts of DNA that can be extracted, amplified using PCR, and then analyzed. Mitochondrial DNA is often used because there are usually more than 100 copies of it in one cell compared with only 2 copies of nuclear DNA. And then here's this example where they found some bones in Russia, and so they used the mitochondrial DNA to identify the bones by comparing it with a living relative. So they found these bones, and then they looked at the mitochondrial DNA of this person.
Okay. Yeah.
Okay, cool. So first, explain why there are usually more than 100 copies of mitochondrial DNA in a cell, but only 2 copies of nuclear DNA.
Is it because the cell has like 2 copies of each chromosome, so not like?
Yeah.
Oh, okay.
But and then mitochondria?
Yeah, and then like because like per cell, like, but like on like there's like only one nucleus.
Yeah.
So not. So
Yeah,
that's
so
probably like a lot.
so yeah, so you're right about the two copies, because you have two copies of each of your chromosomes in your nucleus, but you can have there's no restriction on like how many mitochondria you can have in a cell. So you can have hundreds of copies basically,
Yep.
because that's like independent of your nuclear genome. Okay.
Yeah.
So all the mitochondria in a zygote come from the egg, not the sperm. List the letters of the people in the family tree who would be expected to have the mitochondrial DNA identical to the mitochondrial DNA of the living relative. Okay, cool. So what do you think?
C. So maybe C.
Yeah,
A.
perfect. Yeah, that's that too.
A. F. So
Yeah.
and then C. Oh, and
Yes.
the E and D.
Yeah, that's absolutely correct.
A, F, C, and G. Okay, yeah.
And not B.
Because not connected to A.
Yeah, exactly. And the reason why D and E have it is because they're inherited through the mom, and then the circles are the females. So
Yeah.
okay, I think that was pretty straightforward. Cool. Spraying insecticides on the wall. By the way, does it help if I read it out? Like, I don't have to read it out if you don't want me to.
I think we need to read again because we just.
Okay. Okay. Cool. Okay, I'll read it out then.
Thank you. Sorry.
No, that's fine. I just didn't want to read it out if you. Yeah. Okay. So, spraying insecticides on the walls inside houses is the main method of controlling a species of Anopheles mosquito in rural India. A number of different insecticides have been used. Malathion, or actually I don't know how to pronounce that, was the main insecticide used for many years. In 2005, the newer insecticide, deltamethrin, was used instead, and the use of malathion was discontinued. A lab study was carried out using mosquitoes collected from 2 sites in India. The percentage of mosquitoes killed by malathion and deltamethrin was estimated. Okay, cool. So, what can you tell me about the difference between the effectiveness?
So like obviously I can't say there's some like right one is more effective.
Yeah. Perfect.
And also so the percentage of mosquitoes killed by the right one decreased in the top one.
Yeah, absolutely right.
And one more. Oh, I could find like number of mean, like both of them, like Malthino and.
Yeah, I mean you could do that as well. But I think an easier one is just that overall, if you look from 2005 to 2007, the malathion one just increased.
Yeah.
That's an easy one, I think. Okay, cool, that's 3. And then the researchers concluded that at Jamnagar, the mosquitoes had evolved resistance to deltamethrin. Explain how the mosquitoes evolved resistance. Okay. Do you have any ideas? I'll go back up to the table. Maybe.
How must be like genetic change, like genetic.
Yeah, no, you're right. Yeah, a mutation.
Okay, mutation.
Or just variation. Yeah, I mean maybe it didn't evolve, at this very moment. Maybe there was already like a few individuals that had the resistance gene.
Okay.
So how do you think it would become more common in the population?
Maybe like more survive, like.
Yeah, exactly. Yeah, that's it. Yeah. So if those survive, then there will be more individuals over time that have the resistance gene because they can have more offspring, right?
Mm-hmm.
Okay, cool. And so that means that the frequency of the resistance allele will go up, right?
Yeah.
Okay. I think you got it.
Oh, okay.
Oh yeah, you can mention, this specific one is called like directional selection because you have so the delta methrin is a selection pressure. So yeah, it just okay. Oh, and also it doesn't ask for this, but that's why here, if you see the decrease, that could be why, because there's some resistant mosquitoes that have proliferated in these 2 years, so it's not going to be as effective.
Okay. Yeah.
Cool. So explain how the data show evidence that the use of malathion was discontinued after 2005. Okay. So what do you think? So you can see that it increased, right?
Please, yeah.
So why would that mean that the malathion was discontinued after 2005?
Because the resistance to malathion is no longer like effective.
Yeah, exactly. Yeah, so it's not a selective advantage anymore.
Mm-hmm.
Okay. Is that okay?
Yeah.
Okay, cool. The resistance of mosquitoes to malathion was found to be due to a difference in the shape of one enzyme. Name the type of variation controlling malathion resistance in the mosquito population. So this is classic, you either know it or you don't, what you call it. Okay, so this one is trying to get at the fact that if you can have variation which is like qualitative, so there is a trait, for example, you can have something which is you either have it or you don't, right? So for example, in this case, you're either resistant or you're not. Like there's no kind of halfway resistance, right? Versus something which is quantitative. So like your height is always the one that everybody mentions. So those two are called continuous and discontinuous. But yeah, it's one of those things that you need to know the name of. Otherwise, yeah, but it's fine. It's just a point, so it's not that important, I think, if you miss it out. Although, always good to know. Okay, should we move on, or do you have any other questions about that? Okay. Some students suggested that resistance to malathion could be due to a gene with two alleles. They proposed that the allele for resistance to malathion would be dominant to the allele for non-resistance. Okay. Using this assumption, the data in table 4.1 can be used to calculate the frequency of resistant mosquitoes and the frequency of the allele for resistance in the mosquito population. Use the Hardy-Weinberg Principle to calculate the frequency, P, of the allele for resistance in Jamnagar in 2005. Okay. So do you know the Hardy-Weinberg Principle?
No.
Okay, no worries. So this is a way to calculate the frequency of a dominant and a recessive allele.
Okay.
So I'll write up the equations and then we can go through how I got that. So in this case, this is true because if you only have these two alleles, then you're either dominant or you're recessive, right? So the probability that, yeah, so these are your only two probabilities.
Yeah.
So that's why it's one. Okay, so that makes sense. And then the other one is this one. Oops. Okay. Cool. So would it be helpful to go through like how to get that or not?
I think I need to explain.
Sorry?
I need to understand because I didn't know what they come from.
Okay, that's fine. Cool. Okay, so if you have a new zygote, and just think of, you know, your alleles. So for example, you can have a or not resistance, sorry, just like a dominant and a recessive. Let's just do that, okay? So the capital one is the dominant, this one is the recessive. And so you need to have, you know, you get one copy from your mother, one from your father. So if you draw a tree, for example, you can either have your dominant or your recessive trait in a zygote, right? So if we call the probability of this P, just for the ease of this one, and this one Q, and then if your zygote has the dominant allele, then one dominant allele, sorry, then once again, you can either have your dominant or your recessive, right? And same goes for the recessive one. Does that make sense so far? Okay, and then we can label this P and Q as well. P, Q. Okay. So to get the probability that you have a zygote in which you have the dominant genes, you times these branches together, right?
Okay, so get it. So P squared. Oh. Okay.
Yeah, and then if you go through here, then PQ, and if you go like that, so those will be your heterozygotes, and then Q squared is your recessive, your homozygous recessive. And then all of these must add up to 1.
Yeah.
And then, yeah, that's how you get it. So, does that make sense?
Yeah.
Okay, perfect. Wait, let me just quickly label those so it's easier to. Okay, cool. So what do we know? So we know, so we need to, sorry, so going back to the question, we need the allele for resistance. And we already know from here that the resistant gene is dominant, right? Okay, so we're only looking at Jamnagar 2005 for malathion, right? So if we go back up, that's this one, only this. Yeah. So this percentage refers to your susceptible mosquitoes, right? Because these are the ones that were killed by the insecticide. And if your resistant gene is dominant, then that means that if you're susceptible, you must be homozygous for the susceptibility trait, right? Because if you have one of the resistant copies, you're resistant, right? Does that make sense?
Yeah.
Okay, cool. So the only genotype that you can have if you're susceptible is small r, which is Q squared.
Yeah.
Right? So Q squared is 76%. So if I was to put that into probability form, that would be 0.76. Does that make sense?
Yeah.
From the data. And then if you square root that, what was it? 0.87, I think. But let me check. Yeah, 0.87, okay. And then you can plug that into our first equation.
Oh, so 1 minus 0.87. Okay.
Yeah, so that's just exactly as you said. So 0.13. Okay, is that okay?
Mm-hmm. Yeah.
Does that make a bit more sense now? I think it's easier if you sort of go through because, I mean, yeah, sure, it helps if you memorize it, but like it's good to know what each of these terms mean, right?
Yeah.
Okay. Should we go on? Or do you have any other questions about this?
No, I don't. No, I don't think so.
Okay, cool. You okay?
Yeah. Okay.
Okay. So the land area of Italy is composed of forest, grasslands, built-up areas, and inland water bodies like lakes and rivers. Table 5.1 shows changes in the area covered by these environments over a 15-year period. Okay. So, what can you tell me about this table? I mean, the total area hasn't really changed, so there must be some, yeah.
Okay. So decades of like forests.
Yeah. Yeah, so that's all I was looking for really, is just more forest. And you need to take that area away from something. So decrease in agricultural fields. Okay. So why do you think none of these, as they are here, it can be referred to as an ecosystem? So what do you think this term means? Like, if you remember back, this is a table from a land area of Italy, right?
Yep.
So what does this forest term mean? It doesn't just mean one forest, right?
Yeah, like.
Yeah, exactly. So why do you think that's not an ecosystem? If we just look at the forest for now.
Can I ask you something?
Yeah, of course.
Because I'm not sure about ecosystem. Like.
Okay. That's okay. No, that's fine. Don't worry about it. That's what I'm here for. So an ecosystem is so it's like let's say it's like a collection of populations of different, plants and animals, and they all interact with their environment. So the environment is also part of the ecosystem. So they're called like abiotic factors. So that would be, everything from the weather to the soil, to the nutrient composition and stuff like that. So basically what this question is trying to get at is that an ecosystem, like, it only exists because all of its components are interacting with each other, right?
Okay. Yeah.
So if you have like a population of I don't know, like eagles or whatever, or maybe not eagles, but like, I don't know, sparrows in the north of Italy, they don't necessarily interact with like sparrows from the south of Italy. They can be the same species, but they don't really have much to do with each other, right?
Okay. Yeah.
So they're like separated. So the idea is that an ecosystem is like a system that is like a unit. So it's like a defined area. It's like self-contained. It can it works as a system by itself, so it doesn't need any like external factors to be supplied. So what this question basically is saying is that if you have like just, you know, all of the, this one is I think the easiest one to understand, like for example the lakes, you have like several different lakes all over Italy, right? But they're separated spatially, so they're not one system, right?
Yeah.
That's all. Sorry, that might have come out a bit more convoluted than it actually needs to be.
Okay, I think I got it.
Yeah, you sure?
I think so.
Okay. So like, I don't know, you know, a cow from the Alps is never gonna eat the grass, you know, in Sicily. So
Really?
then it doesn't really have an effect on that part of the country, right?
Oh yeah. Okay.
Okay, that's all.
Mm-hmm.
Okay. The increase in area covered by forest in Italy has occurred because farmers have cultivated less of their land or have abandoned their farms altogether. Describe how this change in land use may affect biodiversity. You should consider the 3 different levels of biodiversity in your answer. Okay, so do you know what those 3 levels are?
No.
Okay, no worries, that's fine. They're defined to be on 3 levels. So if you think of, I think an easy way to remember it is like if you think of small to big. So you're looking for variation, right? So variation. So you can have genetic variation.
Okay.
Then you can have variation in species. So that could be, you know, the abundance of a species.
Okay.
So like how many organisms there are of a species in an ecosystem, or the number of species in the ecosystem as a whole. So you can have like a more complex food web, for example.
Yeah.
And then the biggest one is the variety of like the ecosystems themselves. So Italy is actually a really good example, you know, because they have everything there, you know, from like mountains to you know, more Mediterranean sort of climates, and you know, so that's basically what it's trying to get at.
Okay.
Does that make sense?
Yeah. Yeah.
Okay. Cool. 2 large mammals native to Italy are the grey wolf and lynx. So that's the grey wolf and lynx.
Okay.
And then here's a bunch of facts. So in 1971, there were fewer than 100 wolves in Italy. Lynx were extinct, and wolves received protected status in Italy. And lynx were reintroduced to Switzerland, which is next to Italy, and also later on to Slovenia, which also shares the border with Italy. And then in 2014, the wolf numbers in Italy has increased, but also previously lynx were thought to be extinct, but in 2014 there was 25 of them. Okay. Suggest what actions may have been necessary at local, national, and global levels to contribute to the successful conservation of biodiversity of Italy's large mammals. So what do you have any ideas?
So like stop hunting.
Exactly, yeah, that's pretty good. Anything else?
Feed them, like.
Yeah, that's really it. So yeah, so as it showed on the top, you know, that's why there's more forest, was because, like, you know, if you're a farmer, you're not just gonna give up your farm, right? Like, you need some sort of incentive. So yeah, that's one of them. And then there's a bunch of other ones that you can say, like for example, the fact that, you know, like lynx were extinct in Italy. You can maybe have some sort of agreement with like Switzerland or Slovenia, you know, that I don't know, you're gonna like if you have a habitat, like a big forest, let's say, across the border, that you are going to agree to, leave those alone, so that you don't, cut the habitat in half, for example, just because you have, human borders there, for example. And a few easy ones, like zoos or national parks and stuff like that.
Yeah.
Okay. Okay. And also, yeah, I think another easy one is just like, if you care about, conservation, like you will monitor the number of species, and that's good because then you gain information of like what you need to do as well. So if you don't do that, then you're not going to save, yeah. So that's okay. Cool. Okay. Menstrual cycle question. Okay, so these are the levels of these 4 hormones during a cycle.
Okay.
So the first question is about luteinizing hormone. So it's asking why you have this peak here. So do you know anything about that?
Not sure.
Okay, that's fine. So do you know what follicle, like FSH and LH does?
Nope.
Okay, that's fine. So essentially, I think it may be easier if I get a diagram up. I'm not sure. I'll try and explain this quickly as I can. I won't go into that much detail actually here. There's no point. Like, it's a mark question. So essentially what happens is that estrogen, when it's at lower concentrations around here, it has a negative feedback on the pituitary gland, and the pituitary gland is where luteinizing hormone and the follicle stimulating hormone get released. So can you see that's why it goes down as well, and then LH is like just sort of stagnating. But when estrogen builds up, and then eventually, can you see like there's a little bit of a delay here between these 2 peaks? So what happens is that once it hits like a threshold, then it like all of a sudden has a positive feedback on the pituitary gland, but only if it's at higher concentrations. So it like causes a surge in luteinizing hormone, and actually even in FSH you can see here. So yeah, that's why.
Okay, high concentration.
Yep. Okay. So the diagram shows that the woman didn't become pregnant during this cycle. So can you tell me how we know that?
Is it because like the progesterone? Is it
Yeah.
concentration?
Yeah, exactly. Yeah. So yeah, after ovulation, you have the corpus luteum that produces estrogen and progesterone, and if your egg doesn't get implanted, then yeah, so eventually it falls, and yeah, you get your next cycle starts. Cool. Okay. The combined oral contraceptive pill contains estrogen and progesterone. Explain how this combined contraceptive pill works to prevent pregnancy. Okay, so we can work through this based from the first answer. So what do you think?
It's like
So
it's like maybe like acts as like pituitary gland, so like maybe produce hormones, like not like real one, but
yeah, so does it help if I tell you, so FSH is follicle stimulating hormone. So normally what it does is, as there's more, and if you can see here, as there's more and more follicular stimulating hormone, there's something called the Graafian follicle, and it matures. And what happens after that is there's ovulation that happens.
Okay. Okay.
So, how do you think taking estrogen and progesterone can help to prevent that from happening?
So, how is it? Like maybe HSA, no, FSH.
Okay, so what do we know? What do we know from just the first question? Do you remember? So how does estrogen work again on the? So what does it do in the beginning?
Increase RH. No, I mean,
It's okay. Take your time. Don't worry. Like, so
estrogen. If
yeah.
estrogen like has high concentration, I mean, yeah, high concentration, RH also got high concentration.
Okay. Yeah, but if you take the pill, then you're not gonna have an increasing amount of estrogen, right? You have a constant.
Yeah.
So what do you think? What kind of effect do you think that has?
So we're not
Based
gonna
on what?
have high concentration of LH.
Exactly. Yeah, that's exactly it. So as long as estrogen levels are sort of low-ish, it has an inhibitory effect on these two hormones. So you're never gonna have your spike, and you're never going to ovulate.
Yeah.
Okay, that's it. You got it. Okay. Suggest why some women take the combined pill for just the first 21 days. Okay. So.
Oh, because it's natural cycle.
Yeah. So why does the blood, like the shedding, happen in the first place? If we go back to here, like, you know, towards the end of the cycle, you can see that the progesterone levels are quite low, right?
Yeah.
So if you're taking the pill, then you have a constant source of estrogen and progesterone, right? So if you don't stop taking it, you're never gonna shed your uterine lining, right?
Oh yeah, true. Okay.
Yeah, so that's exactly why. Yeah. Okay. So some women have a contraceptive device inserted under their skin. This releases hormones into the blood and can last for up to 3 years. Suggest one advantage of using such a device rather than taking contraceptive pills. Okay.
Oh, so you don't have to take pills anymore, like not every time.
Yeah, perfect. That's it. That's one of them. Yeah. I mean, you don't need to write any more than that, but do you have another idea?
And like maybe like you go like steady concentration of hormones, like
Yeah.
every time, like not too high, not too low, something.
Perfect. Yeah, that's it. So that also means, you know, that obviously you wouldn't have as much side effects as well as you may get from the. Yeah, but you got it. That's great. Okay. Should we move on? Actually, I don't know. Like, do you? Sorry? I mean, I don't know. Like, how long can you stay, or like how? I'm not really sure if
not the last one. But...
How's it going?
Okay. It's going good, thank you. Hi.
Fantastic. I think what we'll do is we'll kind of sort of wrap it up about now. And
Okay.
Just because
we just got to the end of a question, actually.
Perfect. And we'll just as obviously doing it interactively it takes a lot longer, of course, doesn't it, than going through. But this now, Kaneko, going back and watching the video of you going through it as a fast pass should work quite nicely, I think.
Hopefully, yeah.
We'll see.
Yeah.
Brilliant. Okay. I'll chat with you in the Slack room then.
Okay, sure.
What we'll do as well is, I mean, you're not. When are you coming back to Cambridge?
At the end of September.
End of September. Fantastic. Okay. So at some point, I think we start with biology. Biology is in the middle 2 weeks of the month. So it's in October, it'll be in the week beginning the 9th and the week beginning the 16th. And there'll be at least two of you. So you'll be able to sort of kind of share out stuff there between you guys. So
sure, that sounds really good.
I'll chat with you in the Slack rooms. I was listening to some of it. It sounded great.
Okay.
So well done. And it was very clear. And I watched a little bit upstairs and I could see it's beautifully clear and crisp. So well done. So I'm going to just stop the